Well, welcome. This is our first in-person kickoff for the grand rounds. So thank you all for coming. I really appreciate it. And we picked a beautiful weekend to have an outside speaker. So I am delighted to introduce Frankie Garofalo, depending on the day, to do our introductions. So Frankie.
Thanks, Dr. Schnapp. Hi, I'm Frankie Garofalo. I'm a third-year internal medicine resident, and it's my honor today to introduce Dr. Carol Prives as our Department of Medicine Grand Round speaker. It's safe to say that her work has fundamentally shaped our understanding of cancer biology and altered the course of cancer research, beginning with her discovery of the P53 tumor suppressor. I first met Dr. Prives 10 years ago during my freshman year of college. I was interested in cancer research and reached out to see if she had any room for undergrads in her lab. Luckily, she did, and I started right away. Dr. Prives attended McGill University for her bachelor's degree, achieving first-class honors, and remained at McGill for her PhD in biochemistry. She then underwent postdoctoral fellowships at the Albert Einstein College of Medicine and the Weizmann Institute in Israel. She remained at the Weizmann Institute as an associate professor until 1978, when she began her lab at Columbia University, and she has remained there since. She has been given numerous honors, including being elected to the National Academy of Sciences for her excellence in research. She's also had a fellowship at the American Cancer Society. Additionally, she was the chair of the Department of Biology at Columbia from 2001 to 2004. Dr. Prives has published nearly 275 scientific articles and still leads an active research lab at Columbia. Her talk today is entitled Travels with the P53 Tumor Suppressor Network. Please join me in welcoming Dr. Prives to UW.
Thank you very much, Frankie. I don't know how this happened, but when I was invited to give this talk, I thought this sounds interesting. It's my second Grand Rounds talk, and the first one, I can't remember where it was, but I assumed I'd be standing around a patient's bed with, a clipboard and somehow talking about my research in some completely irrelevant way to the patient. But then it was explained to me this is actually a real talk. So I was invited to this. I accepted. I thought it would be interesting, and only a couple of days ago, because for some reason I started to click on the people who were going to be on the thing, and I suddenly recognized Frankie. I realized, then I saw it. Then I realized how this happened. So anyways, it's very nice to be here. I think my talk is a bit too long, so I may be fast forwarding through some slides. Got the impression I was supposed to give a little bit of personal background as well, so I'll be weaving that into the talk as well. Yes. These are my conflict of interest disclosures. Okay. All right. So as Frankie said, I've been working on, well, I don't know if she actually mentioned the gene that I work on and the protein, but it's P53, very famous if you're a cancer researcher. It's known to be a major tumor suppressor. The story of how P53 was discovered and then was considered for the first 10 years after its discovery to be an oncogene is very interesting. But about 10 years, was discovered and identified in late 1970s, 1979, took about 10 years for people to realize it's not an oncogene, it's a tumor suppressor, but it turns out it's both. And I'll be explaining that to you. As a tumor suppressor, we know it's highly frequently mutated. In terms of, I think, genetic proof that this is a tumor suppressor comes from the Li-Fraumeni syndrome patients. These are patients with a very high frequent disposition to a certain set of cancers well before the general population. And these people have been inherited a germline mutation in P53. We also know experimentally that if you knock P53 out of a mouse, those mice will get cancer with 100% frequency. It was first discovered associated with a a tumor virus, SV40. I'll tell you a tiny bit about that. And we now know that many viruses have evolved to abrogate the suppressive functions of P53. And wild type P53 actually can suppress the ability of oncogenes to form culture cells. So P53 sits as a hub. It's been actually analyzed computationally a lot now at sort of a central hub where it receives many different kinds of stress signals. I wonder if I can, I hope this can be seen. Yes, it can. I guess it can, yes. So oh, it's over here. Yeah, okay. So basically, it's a sequence-specific DNA binding protein. That's a transcriptional activator. I'll tell you a bit about the history of how we discovered that. They receive signals from many different kinds of stresses, transmit those signals to genes that cause many different cellular outcomes consistent with tumor suppression. It's held in check by two proteins, MDM2 and MDMX. And it's at least the relationship between P53 and MDM2 is a negative feedback loop. P53 induces MDM2, which then suppresses P53. This circuit has to be broken, and it is broken by these various stress signals. And DNA damage is probably the most well-studied stress signal. Many different kinds of DNA damage activate P53, and they cause different outcomes. The most well-studied is cell death. But now, as you can see, from the result of hundreds of researchers all over the world, it's a very, very complex network. And I won't get into it. I could probably spend the whole seminar talking about this, but it's got multiple regulators and outcomes and relationships. It's very, very complex. We learn constantly to this day that it's very complex in another way too, in that each of our tissues actually has a unique P53 transcriptome. And so in fact, and I find that fascinating. It's a very, very complicated protein. And as you can see here, when you look at an analysis of the TCGA, the Cancer Genome Atlas, that it looms above all the other oncogenes in terms of the frequency with which it's mutated. So here's my life story, of which you heard a little bit from. I was born and raised in Montreal, and I did postdocs, 4 of them actually. And the last one was at the Weizmann Institute, where I stayed and eventually got a faculty position. And I did 2 sabbaticals, one at the NIH, 3 sabbaticals, one at the NIH many years ago with the late, great George Curry, and then with the late Jerry Hurwitz, and more recently with Bruce Stillman. So how did I get into studying P53? So as I said, when I was in Israel, I worked on SV40. And at the time, SV40 was in some ways, in terms of people really wanting to understand how genomes work, it was kind of the only game in town. It was gene cloning had just been discovered and analyzed. SV40 actually was the genome for which the first restriction map was ever generated. And people studied it also because if you introduced SV40 virus into mouse cells, the mouse cells became transformed into oncogenically. If you put them into newborn baby mice, the mice came down with tumors. So the tumorigenic part of the virus was this region over here that encoded the large T antigen, which is an amazing protein. It's not only a transforming protein, it's also a very complex DNA replication initiating protein. And in 1979, 2 groups, one of them was Lane and Crawford, and the other was Arne Levine, discovered there was another protein associated with P53. When I was working on SV40, I thought I'd really like to learn how it replicates DNA. So I did my first sabbatical with Jerry Hurwitz, and then later with Stillman and this person, Tom Kelly. These were the 3 giants who basically worked out the whole mechanism of how SV40 DNA is replicated. And I was very interested in that. And I realized when I did my sabbatical with Jerry Hurwitz, I have to be seriously insane to compete with these groups that had armies of people purifying proteins and everything. but they were completely oblivious to the fact that there's this other protein associated with T antigen that I thought was pretty interesting. And I got very lucky when a fantastic virologist, Lois Miller, contacted me and asked if I was interested in actually expressing T antigen in a baculovirus. And that was really my entry into the into the field. And so another person whose picture should be on this slide was Bert Vogelstein. He was a very, to this day, a very distinguished cancer geneticist who has identified many proteins. And it was really his discovery that wild type P53 is frequently mutated and is a tumor suppressor. And Bert had given us not only wild type P53 gene, but also very mutated versions of P53 that turned out to be the most common mutated versions. And when Jill Barganetti came to my lab, she ended up doing a footprint, a DNase footprint. People don't do these very often anymore, but it's a fantastic way of looking at how a protein binds to DNA. You can really see what regions of the DNA are protected. And one of the most striking results she had was that wild type P53 could do this, but these tumor-derived mutants were completely unable to bind to DNA. So we published several papers. George Farmer in my lab put all this together and set up an in vitro transcription system. So we were able to show that P53 is a sequence-dependent transcriptional activator. Other people made these discoveries as well using different systems. And then we did some other studies. I won't go through all this, but over the years, we developed a lot of other, I think, potentially interesting observations about the protein, including the fact that it can slide on DNA, and including the fact that the way that these, the different, P53 is a tetramer, and that's something I'll be mentioning in a moment, the way that the different molecules in the P53 tetramer interact with each other dependent on other regions of the protein. So we did a lot of work in that area. You can see this is a map of P53 and the mutations that are found in it. We know it's a little more complicated than that. When it was first looked at, it was assumed that all of the mutations were clustered in this region, but actually they spread throughout the protein, but among them, there are what we call these hotspot mutations that occur with very high frequency. And I believe that these are actually oncogenic. They're not just neutral mutations. And I'll talk more about that in a while. Just going on, when I told you that P53 is negatively regulated by MDM2, this very wonderful postdoc, Shouyan Shea, came to my lab. And she phosphorylated P53 and showed it couldn't bind to MDM2. And that was an interesting observation because around that time, it was discovered by other groups, Moshe Oren, Karen Voustin, and a group in Japan, that MDM2 can degrade P53. And so what we were able to hypothesize was that when P53 is stressed, it becomes phosphorylated, and therefore it can't bind to MDM2, and that's why it gets stabilized. And that's what happens in normal cells. The levels of P53 are very low in cells unless stress signals activate P53, which leads to its stabilization and activation. So we then, as a part of the work that was going on in the lab, got very interested in MDM2 itself. And we did some studies thanks to Moshe Poyarovsky and Yen Zhu and Hen Katz about various aspects of MDM2 as well, how aspects of its structural features, and the fact that MDM2 could target P53 much more efficiently when P53 is a dimer instead of a tetramer. And I'll come to that in a moment. So Hen over here was the lab mentor of Frankie. And when I realized that it was Frankie that was responsible for my being here, I actually contacted Hen, who then sent me some nice photos of Frankie showing her poster on the work that she did as an undergrad in my lab. And that's Frankie. This is another undergrad, Jasmine, and this is Hen over here. sent me some other pictures too, but they're kind of funny pictures. So just a little more about MDMX, and I'll start talking about recent data. These are just some recent studies we've done. So we believe, and this is a little more complicated, that in cells that don't have wild type P53, and when I said P53 is very frequently mutated, I would say that about half overall, of course, it varies enormously with the cancer from very low to very high. About half of all tumor-derived mutations in P53 are found in human cancers. It varies from like almost 100% in ovarian cancer to very low in, let's say, cervical cancer. But those tumors have MDM2. And we have evidence, and so do others, that in tumors that lack wild type P53, MDM2 is actually regulating oncogenic processes. Very recently, we've just published a paper, it just came out, showing that MDM2 regulates actually processes associated with metastasis. So, you know, searching for compounds that affect MDM2 function, I think, is a very valuable direction for researchers to take. Okay, so again, this is a diagram, somewhat old, of P53. These are the hotspot mutations. And it is believed, and it's controversial, that whereas wild type P53 has all these activities that I described before, cell death, cell arrest, senescence, it affects metabolism, it affects the immune system and DNA repair. Mutant P53 has its own set of activities in cells that lack wild type P53, full-length, highly frequent mutant versions of P53. They actually, and this has been well documented, have a lot of activities associated with oncogenesis. They can increase survival, they change the metabolism, they stimulate migration and invasion, and I'll talk about that as well. I said it's controversial because this is considered what we call a gain of function, a gain of oncogenic function. And I think the data for that are extremely strong in cell culture. I'll show you some examples. And I think the data in mice are very strong. So if you knock P53 out of a mouse, the mouse will get lymphomas and sarcomas. If instead of knocking out and having no P53, you introduce into the genome one of these highly frequent mutations, the mice get metastatic carcinomas. So that's a true gain of function. And and yet it's been challenged, which is sort of interesting. All right, so how do these mutations and this is why, because it's when you don't have wild type P53, you lose its wild type functions. And this can happen either from losing P53 or in some cases having mixed tetramers where there are both wild type and mutant proteins in the same tetramer. And it's been shown that mutant P53 represses the wild type P53 by a dominant negative activity. But then there's this acquisition of what we call neomorphic oncogenic characteristics. And I hope I can convince you we can show that. So here are three lines of evidence for mutant P53. First of all, mutant P53 is present at much higher levels in most tumors, in both humans and in animals, than you would ever find with wild type P53. And in fact, there's evidence that if you lower the levels of mutant P53 in those settings, you lose the oncogenic characteristics. So high levels of stable protein are associated with these activities. If you overexpress mutant P53 in null cells, and I can show you that, or not even overexpress it, just endogenously express it, you can get activities, but it's been shown to cause tumor formation in xenograft models. And as I said before, there's a difference between knocking out P53 in a mouse and knocking in a tumor-derived mutant of P53. So this is the first story I'll tell you. And it's linked to actually a mutation that has occurred in a very small set of Li-Fermani families. So as I said before, these are individuals who have germline mutations in P53. They've actually formed an association, which I think is wonderful, because so that they can be a support system for each other, they can share information, their oncologists can share information, and so on. And they have a meeting every few years. And I and several other basic researchers in the field have been fortunate enough to go to these meetings and meet these patients. It's very, very inspiring, and sort of their courage is amazing. At any rate, so as I said, and this is Frank who was involved in this, we discovered that MDM2 prefers to degrade the dimer forming version of the dimeric form of P53. So P53 exists. There's evidence that in normal unstressed cells, a lot of the P53 is a dimer. And then after the cells are stressed, it becomes a tetramer. And so there are mutations in P53, in the region of P53 that is needed for its oligomerization, that actually force it into the dimer conformation. And when I was at one of these Li-Fermani meetings, I was approached by a patient who just was talking to me, and he offered to provide some of his fibroblasts and those of his family. And it turned out that he had a mutation in the dimerization domain. And so starting with the original observation that was made by Hen and with Frankie's help about that MDM2 prefers dimers, we started to study aspects of dimer forming P53. So another undergrad in the lab, Josh Choi, I don't think he overlapped with Frank. He might have come right after her, but he stayed for a few years in the lab, also worked with Hen and a bunch of other people in our lab. And then we had a great collaboration with Dongfang Li in Texas at UT Houston. And I'll explain why he did this and why it might be interesting for clinicians here to think about things you can do with the fibroblasts of patients. And these were other collaborators. Actually, these two physicians, Josh Schiffman and Paul Wong, who both work with Li-Fermani patients, they worked with this family to provide us with fibroblasts. So let me just remind you again that P53 is a tetramer when it's active, and it's considered, to exist. It's basically, we believe that dimers are formed co-translationally, whereas tetramers are formed post-translationally. And work from, what's her name? I can't remember it right now. At Harvard, Galit Lahav, she's actually done some beautiful work showing that in unstressed cells, P53 is mostly a dimer. After you DNA damage, the P53 converts into a tetramer. And I'll show you, we can show that too. This just brings me back to tell you about Li-Fermani syndrome. This was Fred Lee and Joseph Fermani, who I think is still alive. Fred Lee died a few years ago. They identified these families, and this led to this understanding a lot about this syndrome. This is actually Li-Fermani families get a pretty similar, I think, distribution of mutations. There is this one very interesting mutation that affects some aspect of the oligomerization domain. And then the mutation we look at is, whoops, I guess, let me see, go back. Yeah, this is ours over here. It's a very, very rare mutation. There might be like 50 patients or so worldwide, but it's the only dimer forming mutation that is known in this patient cohort. So these are the systems, as I've mentioned, that Dongfang in Texas, he established a system, actually as a postdoc, where you take fibroblasts from patients and you then de-differentiate them with the Yamanaka protocol. These are these 4 genes that turn them into basically induced pluripotent stem cells. And then you can re-differentiate them with different factors into the precursors of different tissues. In this case, he generates osteoblasts. And he showed that and osteosarcoma is a very common disease that Li-Fermani patients get. It's one of the common ones. And he showed that, in fact, the characteristics of the osteoblasts of these patients were pathologically very similar to osteosarcomas that you find in patients. And basically, unfortunately, I think, yeah, you can see it here. We had 2 patients and 1 unaffected sibling that doesn't have a mutation, which is a very useful because it gives you a better control. It's not perfect, of course, but it helps. So we were able to compare. And I'm not going to show data with these patients, but every experiment I'm going to show with this system we saw also in these derived osteoblasts and even the mesenchymal stem cells from these patients. So what we've done, this former MD PhD student in the lab, actually took UTOS osteosarcoma cells and he manipulated them with CRISPR to generate knockout cells, heterozygotes, and this would be like the Li-Fraumeni system in normal cells, and then basically knock in, and he knocked in the same mutation that this family has. So we had a very well-controlled set of lines. And so actually, I just want to point out that this is also a very good way to show, I think, genetically gain of function. You can start with wild type, nothing, and then mutant. So you can really see if there's anything different about these mutant cells, it's a gain of function. So first of all, we were able to show that if you treat cells with a common chemotherapeutic etoposide, this mutation will only form dimers, whereas the wild type will form tetramers as well. And very satisfyingly, you get about half as many tetramers with the heterozygote, and then none essentially with the homozygote. And also, we realized very rapidly that this version of P53 that has only the mutant, and I should say, by the way, I should have told you that when Li-Fraumeni patients do develop cancer, they have lost the wild type allele in their tumors. So they don't have any wild type P53 in their tumors, which must be linked to how they eventually come down with their various tumors. At any rate, the homozygous version of these cells was unable to induce any classical P53 targets. So we went on to do somewhat more genomic analysis. We did RNA-seq and discovered that if they're transcriptionally inert, so this you'd say is like a loss of function. It lost the ability to activate transcription of normal P53 genes that you see either with the homozygous or their heterozygotes. But when we did a ChIP-seq, when you're looking to see what regions of the genome are bound by the mutant form of the protein, it was completely different than what you see with wild type. So we call this a gain of function. It's just new sequences are bound. And this we did pathway analysis. And a lot of these genes that were bound were somehow associated with mitochondrial functions and dysfunctions. So we also did a metabolic profile and using a sequence and discovered that these mutations have what we call a glycolytic phenotype. I think I unfortunately showed this slide twice, so forget that. But the most striking, I think, morphological was that the mitochondria in the cells that had only the mutant in them looked really weird. I know weird is a very popular word nowadays. They looked very strange. And we won't get into that. But they did, as you can see, they do look pretty weird. And it was definitely because of the P53 in those cells, because if you knock down P53, you lost that very strange looking morphological characteristic. And we also knew that, we also knew that you could inhibit, well, no, that's not true. So this is a gain of function. The other thing we discovered was that the cells from these patients, and that's why I sort of like to present this to people who sometimes like to think clinically, were hypersensitive to certain chemotherapeutics, particularly, for example, etoposide, much more easy to kill them and this was true both as the cells of the patients as well as these engineered cell lines that we had. So they were much easier to kill. So what we know about P53 as a transcription factor is that it works as a tetramer. And as a dimer, it's extremely defective. But at least one of the reviewers of the paper said, but can you prove that this response, this hypersensitivity to etoposide doesn't have anything to do with transcription? So we treated the cells with actinomycin D. By the way, the kind of cell death we saw was apoptosis, because you could reverse it with an inhibitor of apoptosis, ZVAD. But what we found was that if you treat the cells with actinomycin D, which really wipes out all transcription in the cells, these cells were still super sensitive to etoposide. So this is a transcription independent response to P53. So that's a gain of function too. And then we thought it had already been shown by Utamal and Doug Green several years ago that P53 can bind directly somehow to mitochondria. How it makes those interactions, we don't know. But the conclusion was that that's one of the ways in which P53 induces cell death. So we thought we'd look at mitochondria, and indeed we found that this mutant binds to mitochondria, and it also can be immunoprecipitated with two genes, with two proteins that are known to regulate mitochondria, BCL2 and BCLXL. And we developed, I think a pretty good case that this association with mitochondria is required for this mutant versions of P53 to produce apoptosis. So several years ago, Andrei Gudkov and his colleagues isolated a compound they called pifthrin-mu that disrupts binding to mitochondrial proteins such as BCL2 and BCLXL. And we found that if we treat the cells with this compound that you could buy from Sigma, in fact, you would decrease the ability of this protein to bind to mitochondria. This is the mitochondrial fraction. And furthermore, there you go. And furthermore, this compound actually significantly reversed the ability of these cells to die when they're treated with etoposide. So I think that strongly argued that the cell death, that these particular leaf-Romania-derived mutation that is hyper, I guess, responsive, let's say, to compounds such as etoposide is actually due to its interaction with mitochondria and has nothing to do with transcription whatsoever. So that's just the take home of this part of the talk. Is a hyperstable mutant, forms dimers only, doesn't form tetramers. It has lots of gain of functions in the sense you see activities in these cells with the mutant, but not either in the null cells or the wild type cells. And Josh described this as a double-edged sword. Losing transcription is obviously pro-tumorigenic, but it also induces apoptosis directly in a transcription-independent manner. And I like to think that if one could set up a system like that with patients, this is sort of like personalized medicine, where you could actually derive, differentiated components, you could then screen them for drugs and see whether you could pick up some combination of chemotherapeutics that could be particularly effective for a specific patient's tumor. Obviously, it's not a totally original idea, but this is, I think, a strategy that is worth pursuing. So for the next part of the talk, which is sort of most of the rest of the talk, it's also a gain of function story. And it has a published component and an unpublished component. So I'll talk about the published component first. This is the work of an MD PhD student that came to the lab, I guess, well over 10 years, because his paper was published over 10 years ago, Will Friedpaster. He's now on the faculty at Dana-Farber. And when he was in my lab, we set up the 3D system where you put cells into laminin-rich extracellular matrix and they form little structures. If they're normal cells, they form structures that resemble the ducts of the mammary gland. But if they're cancer cells, that had been shown by others, like if you take breast cancer cell lines from many different sources, they never form these what we call acinar-like structures that actually have hollow lumens and that have a very beautiful polarized structure. And the cell line he worked with, MDA MB468 cells, when put into this culture, formed very unusual malignant-looking or intermediate structures with a tiny fraction looking normal. And Jill Barganetti had made inducible shRNA against the mutant P53. I should say these cells contain a common mutant P53, one of the frequent ones. If you knock it down, you converted this tiny fraction to a very large percentage of the structures seen in 3D. This morphological reversion to normal was very dramatic. We showed, by the way, that this required the activation domain of mutant P53. And we did analysis in collaboration with others and discovered that a pathway that was very frequently altered in these cells was the mevalonic pathway. This is the pathway by which cells synthesize cholesterol, which is very important for membrane function. But byproducts of that pathway are also very important for the attachment of G proteins that are signaling and growth signaling pathways. So it's very important for the attachment of these, what they're called small G proteins to bind to the membrane. Many other byproducts are important. It's a very important pathway. It's also, as you know, the target of these blockbuster drugs, the statins, that prevent hypercholesterolemia. And there are also drugs that have been isolated with the attempt, I think, also of clinical use, but I think somewhat disappointingly, that block various pathways, including ones that inhibit geranylation. And this pathway is actually regulated by a master transcription factor called SREBP2. And Will hypothesized that mutant P53 stimulates the ability of SREBP2 to activate this entire pathway. Did a lot of experiments to support of that, came up with this model that in cancer cells that have mutant P53, you would predict have very high levels of this pathway. And in the experimental system, this would be driving a very malignant phenotype of the cells, which you would lose if you got rid of the mutant P53. And this was before TCGA, or certainly before you had enough statistical analysis. We collaborated with Annalisa Boris-Dale, who had a lot of data sets from patients, breast cancer data sets, gene expression, and discovered that there was a cluster that had very high levels of the mevalonic pathway that correlated with having mutant P53. And in fact, she even had survival data. And the patients that had low normal levels of this pathway had better survival than those that had these very high levels of these enzymes with the mutant P53. So as I showed before, there is, we think, a reciprocal, almost yin-yang relationship between wild type P53 and mutant, where quite strikingly, those genes that are activated by wild type P53 are actually very often repressed by mutant P53 and vice versa. So we hypothesize that maybe these genes are repressed by wild type P53. And here's where I'm going to fast forward. We discovered this is the case. We had a great collaboration with Scott Lowe, did a lot of work, and I'm just going to zip through them, other than to show that we figured out that the target of wild type P53 that's needed to repress the mevalonic pathway is almost counterintuitively a transport protein called ABCA1 that's known to both export cholesterol from cells, but it also can retrograde transport cholesterol back into cells. Did a lot of experiments to show that. Going through it very fast. Did a nice collaboration on tumor analysis with Scott Lowe, showing that loss of ABCA1 actually predisposes the cells to hepatocellular carcinoma. And we ended up with this kind of a sort of a diagram here showing that this pathway, where we go from SREBP2 to mevalonic gene pathway, is negatively regulated by wild type P53. And as I showed you, it's activated by mutant P53. So very recently, now we move into an unpublished area, we've been trying to figure out if the relationship between this pathway and mutant P53 is linked to two very critical and deadly aspects of cancer, which is basically the propensity of cells to leave a primary tumor, and that as the first step in metastasis. And this is the work of Aja Guzman with collaborations with other people, including Lara Kaufman in our chemistry department. And what Aja has been doing is a protocol where you take cells growing in 2D on plates and you disperse them and you put them into what is known as low adhesion tubes that are somehow treated so the cells can't attach to them. And they form these little balls called spheroids. And then Aja implants these spheroids into a biomatrix that would supposedly represent or mimic the extracellular environment into which cells are normally, tissues are normally present. In this case, she uses collagen or collagen plus matrigel and other various different. She has a lot of she's done a lot of analysis on what is needed to either promote or prevent cells from doing this, from leaving the spheroid and basically invading into the matrix. So this is the experiment. It's a bit laborious, but she's gotten a lot of very interesting information. So here are 3 lines of evidence that you need mutant P53 to do this. And so I'm very convinced myself. First of all, just a classical knockdown experiment where you treat the cells with siRNA, either a single siRNA or a pool of siRNAs, you get much less invasion, and this can be quantitated. Alternately, you can knock out, these are from a cell line called MDA231s. You can knock P53 out of these cells using CRISPR, and you get, again, much less invasion. These are different clonal derivatives. So you see some variation, but they're always much less than the parental cells. And finally, we've knocked into another cell line called HCT116. These are actually a colon cancer cell line, a hotspot mutant called R282W. This has been done because this was made easier because you can use base editing to generate this mutation. So in these cells, we have a knocked-in mutation, a knockout and wild type, just like I told you before. Again, this gives you a very good allelic series to really, I say, confirm or disprove. I think we've confirmed that there is a real gain of function. So in this case, where you knock down or knock out mutant P53, you get less invasion. Here, when you knock in mutant P53, you get more invasion compared to the knockout or the wild type. So you can do this in another way too, which is a little more physiological. We've got metastasis experiments hopefully starting very soon. But this is taking lung tissue from mice and decellularizing it, and then incubating this with your different constructs. In this case, wild type, or not wild type, mutant P53 or the knockout clones that don't have, and look for colonization of this lung tissue. And you get much less if you've knocked out mutant P53. So we've learned some things about the mutant P53 in the cells that maintains the rounded shape. The cells get longer and skinnier if you knock out P53. And the round shape of cells is associated with something called bleb morphology. This is really hardcore cell biology, and I'm not much of a cell biologist, but that in turn is linked to the contractility of cytoskeleton, which is related to Rho-ROCK signaling. And in fact, if you inhibit Rho-ROCK signaling, you get much less invasion with these 2 cell lines, but not with the knockout cell line. You need to have mutant P53 to see this big reduction. And as I said, the mevalonic pathway is involved in this because it provides the isoprenyl moieties that are needed for Rho to attach to membrane and then activate ROCK. And if you inhibit the mevalonic pathway, you get the same loss of invasion that you get when you just simply knock down mutant P53. And more strikingly, you can actually rescue the big drop in invasion by mutant P53 by adding one of the precursors of the isoprenyl moiety, namely geranyl pyrophosphate. So you completely reverse this. So furthermore, we can actually show that mutant P53 plays a role in the actual association of Rho with cell membranes. I'll just show you the biochemical result here. This is the membrane fraction of cells. If you knock down mutant P53, you get much less association of Rho A with the membrane. So the hypothesis we've got is that mevalonic pathway gene expression is repressed by wild type P53, goes up if you lose P53, and you get an even bigger boost if you have mutant P53 in the cells. And we can show that with some, but not all of the genes. So this would be the parental cell, the knockout, and then the knock-in. And in some cases, you get a clearly big increase with the mutant. Not always, but in some cases you do. This is a particularly interesting gene that we've pursued. We've looked, for example, in patients' data sets, and you can see significantly more of this IDI1 component of the mevalonic pathway. Also has a small impact on patient survival, which is hard to see in the TCGA very often. And furthermore, if you generate cell lines when you've overexpressed IDI1, they're much more, well, they're a lot more effective at invading out of these spheroids. So this is the pathway we think we've identified. Mutant P53 is needed for extra activation of the mutant of the mevalonic pathway. One of the, perhaps the key product of this pathway that's needed for the impact of mutant P53 on invasion of cells is providing more building blocks essentially for Rho A to attach to the membrane, activating ROX signaling, which leads to cell migration. So for the last little bit, which will only be a couple, one of the things that we've been struggling with is seeing whether we can find examples in cancer patients' data sets that support the idea that there is a real gain of function by having mutant P53. And this is very challenging. It's, in fact, it was very disappointing to me when we first started looking. I just assumed we would see a big impact. There's not a very big impact on, let's say, comparing mutant P53 in many breast cancer data sets, whether or not the patients survive longer if they've had wild type P53 over mutant. I can give any number of reasons, but we do realize that these patients have been treated with various kinds of chemotherapy and other kinds of therapy. So it's not, you obviously can't do a really clean experiment, and you would not want to. There is one case though, and you can criticize this, but I think it's interesting. And this is the work of Kaushik Raghunath, who's a bioinformatics person in my lab. And he's just published a paper recently where he was looking at, I'm a little bit rambling here, but it'll come together in a moment. He's shown that using an RNA-seq analysis, he's shown that that wild type P53, not mutant, wild type P53 regulates a lot of the long non-coding RNAs in the genome, maybe 1,000 or so at least. And what's interesting is that if you then go into patients' data sets and you look at the relationship of P53 status with these different long non-coding RNAs, first of all, it's very clear there is one. And second of all, it's very different as a function of the tumor of origin. So glioblastomas and colon and breast each have a unique set of long non-coding RNAs that are P53 targets that you don't find with others, and there are very few in common. And this is what people are finding also with coding genes of P53. And then they did an analysis. This is a collaboration with Raul Rabadan at Columbia, who's a very good computational biologist, looking to see if they could tie in survival with these different non-coding RNAs. And yes, there was a correlation of a subset of these different P53 targets with patient survival. But again, it was completely or almost completely unique to each of these different cancers with virtually no overlap in the middle. So that then again tells us that P53 is very complex and very, very tissue specific in the kinds of targets it has. And so this is just some analysis showing it. They're not big differences, but sometimes you do see differences. And now I think the best thing to do is actually look at individual targets of P53 rather than P53 itself. But the most striking example where we can see something is looking at in aggregate hematological target tumors. First of all, in these tumors, having any mutation in P53 is horrible in terms of survival. However, if you now segregate what we call nulls, these would be truncation mutations that are either, they're not making a full-length protein. Their survival is significantly better than if you have a missense mutation. So this is sort of what I think is, I guess, a computational and perhaps clinical evidence that there must be something about having a full-length P53 in cells that's a mutant that is not good. So I'm going to stop here. I want to thank you. This is a not terribly recent, but quite recent picture of the lab. I always like to put in the country of origin of people because science is very, very international. And I think it's very important for people to be able to come to this country and do science here. So thank you very much.
Great, really wonderful journey. It brought back some memories for me because my first research project as an undergraduate was also working on, I was working on RSV and chick embryo fibroblast.
Oh, really?
Way back when. I have not continued that line of research.
It's never too
line.
late.
So I'm going to ask the first question. This last piece of data that you were sharing, if I understand, the same P53 mutation, if it's found in a breast cancer, has a different set of potential downstream
I think so.
targets than
Yes.
in lung.
Yes.
So my question is, do the metastases retain the phenotype of the place of origin, or do they now turn into a
That's a fantastic question that obviously we're not in a position to do that, but a collaborator we have wants to do that in her future. I think it's a fantastic question. To what extent they become really different as they find their way into a totally different target tissue.
How does the extracellular environment
Yes,
influence?
Absolutely.
Its,
fascinating question, a very important question. Yes.
Yeah, a great talk. So you showed in breast cancer, the melanoma pathway correlated with survival or poor survival. Are these mostly triple negative breast cancer? Do you look at the different cell type, subtype of breast cancer?
I don't know anything more about what the kinds of breast, for example, I don't know, what kinds of, breast cancer can be subdivided into luminal and this and triple negative. I know nothing about, whether that, my guess is, from everything we know, is that the triple negative basal is the worst for them. But the question then is, do you have higher levels of mevalonic acid in those kind of tumors? And wait, maybe I have this data here, just with that one. Yeah, try that now. Yes, okay. So this is, okay, here's another thing I learned. It's so obvious, and yet I was very surprised, is that it's not so much, in terms of survival, it's not so much various mutations, it's actually the stage. So this is like old, right? We've all sort of known for a long time that if you stage a cancer, the stage 4 is the worst. But in fact, in these other stages, if you look at this particular gene in the mevalonic pathway, you see that it's the most, I think, highly expressed relative to the other wild type and truncated as you go further, as the stages get worse. I don't know if that is a very small piece of data, but it's consistent with that idea.
Yeah, so my other question is related to the earlier talk, part of your talk. You mentioned the dimer P53 interact with BCLXL.
Yeah.
So from the structure point of view, can you see the tetramer form no longer have that surface to interact with BCLXL?
I don't know. That's an important question. I think structural analysis of P53 is incredibly important. What?
I have another question about that, those really intriguing P53 dimerization only mutant, right? Of course, we'd love to look at the post-translational modification that, that our group looks at. That'd be really interesting. But do we know anything, does that, what does it interact with like P63 or P73? And is that part of?
That's a good question. It's oddly enough, because we've always, many years ago, we may have been the first to show mutant P53 bias. It was a very small paper, but I think it's been picked up by people and done much more rigorously. We never did that, oddly. We could do it, but it's an interesting question.
It could be part of the difference of its
Yes.
functional manifestations.
Human chain.
So in medical oncology, we've developed some small molecule inhibitors that block MDM2.
Yes.
I know we spoke a little bit about this at dinner last night.
Yeah.
Shifting to the P53 wild type population, we've seen responses in individuals who are P53 wild type and have very low levels of MDM2 amplification, copy numbers as low as 6.
Yeah.
Could you speak in your wealth of knowledge to the use of things like Nutlin in the lab in your P53 wild type state, specifically as new drugs are developed in that space that potentially could be improved in their tolerance, pill treatments that are given once a month?
Yeah.
Is that something that you've investigated from, say, Nutlin or other MDM2 inhibitors?
I don't know if we've ever asked the question that way. We published a paper, I should say, a couple of years ago with Brent Stockwell, who is the person who kind of discovered and named ferroptosis, which is a kind of cell death. It's pretty sort of a hot topic these days. By the way, that mutation, the dimer, absolutely does not affected by any ferroptotic drugs. It's not interested in ferroptosis at all. But we showed that MDM2 and MDMX actually facilitate ferroptosis. So the context of which, so first of all, I believe MDM2 does stuff in cells that has nothing to do with P53. But when P53 is in the cell, and especially when the cell's being stressed, whatever P53 does completely dominates any kind of activity MDM2 does. This is not answering your question, but in those experiments, we got a hold of cells with amplified MDM2 and found that they too were very effective at promoting ferroptosis. That doesn't answer your question at all, but I did want to ask you something, because when you brought it up, when you said you don't see amplification, but did you ever look at the levels of the protein? Because there could be, some other E3 ligase. We don't think the MDM2 only regulates itself, its own levels. And so maybe there's still a ton of MDM2 in those cells, even if you don't see it even at the RNA or DNA level.
So in terms of current precision oncology, we use DNA amplification calls in terms of enrollment. And there's no kind of central enrollment criteria that's based upon protein expression or IHC. I think the other component to that is large structural variance as well, which is an open question. I think specifically when you stratify outcomes like in hematologic cancers where large structural variants are there, that might be an important
Be very
component
interesting. That would be an
to pull
interesting.
apart.
Thanks for a great talk. So the invasion assay you showed looked like a great way to be able to take advantage of the biochemistry and the molecular biology, you know, and actually screen for repurposing drugs that prevent metastasis. I think you showed some data. Could statins be used to block metastasis in tumors that are P53 driven?
Yes, yeah, yeah. Yes, but it's a rather labor, and you'd have to figure out how to mechanize it and make it high throughput, right? And it's a little tricky to do that. We've talked about it. It's kind of labor intensive. She literally manually picks these little spheroids and puts them in various biomatrices. I'm sure there's a way you could mechanize it, but we haven't thought about that. But it's a great idea because I do think that's a better way to try to test drugs. What we've found both now, well, we've found it, more than one investigator in lab has shown that these drugs do very little to mutant P53 cells growing on dishes in 2D. They really have to be in some kind of a 3D, much more physiological environment. And I bet in terms of drug screening, if you could set up that in a mechanized way, you get information that you wouldn't get otherwise. I'm sure people are doing that, aren't they? Yeah, they must, like organoids, but I mean in a sort of high throughput way.
Great, well, thank you. I think the take home message is everybody should be on a statin.
I'm on a statin. Actually, people always ask me, you know, is there any clinical evidence? And it's complicated. The only evidence that looks good is liver cancer. If you look at all the meta-analyses, there does seem to be some protective